Hello everyone, welcome to today's session. Today we are going to see class 12 biology chapter 5 molecular basis of inheritance. In that we are going to see DNA replication. So in the previous sections we learned that DNA is the genetic material is responsible for storing hereditary information. But an important question arises. If every cell receives DNA from its parent cell, how does DNA make an exact copy of itself before cell division? Answer is very simple. It's DNA replication. Yes, let's get into the topic. Semiconservative replication. When James Watson and Francis Crick proposed the double helix structure of DNA in the year 1953, so they immediately realized something remarkable, and also they wrote the specific base pairing immediately suggests a possible copying mechanism. This means that because adenine always pairs with thymine, guanine always pairs with cytosine, and each DNA strand already contains all the information needed to produce its complementary strand. Then how does this DNA replicate? Just to imagine now opening a zipper. Yeah, similarly, the DNA double helix unwinds. You can able to see that in the picture here. Yes. So the DNA double helix unwinds. The two parental strands separate. The two parental strands separate. So each old strand now acts as a template. And free nucleotides from the nucleus pair with the exposed bases. Exposed bases according to base pairing rules. And finally, two identical DNA molecules are formed. And each DNA, daughter DNA contains one old strand. That is, this is one old parental strand and also one newly synthesized strand. So what is the meaning of this semi-conservative? Semi means half, conservative means preserve. So each new DNA molecule, it conserves one old strand while synthesizing one new strand. And this is why DNA replication is called semi-conservative replication. So you can able to see in this figure, yes, parental DNA opens and also two strands are separating, right? And each strand, it acts as a template and it forms complementary nucleotides, right? And they are added to the strand, right? And finally, two daughter DNA molecules are formed. And each daughter DNA contains one parental strand and as well as one newly synthesized strand. And this is a most important point in the diagram. Next, we are going to see the experimental proof, Meselson and Stahl, 1958. And why was this experiment needed? See, Watson and Crick proposed semi-conservative replication only as a model. And scientists, they wanted some experimental proof also. So this proof was given in the year 1958. In the year 1958 by, who was the scientist? Yes, Matthew Meselson and Franklin Stahl. Matthew Meselson and Franklin Stahl. So their experiment, it became one of the most famous experiments in molecular biology. So what they did, in the first step, they were growing this E. coli, that is the Escherichia coli, in heavy nitrogen. The scientists, they grew the E. coli bacteria in a medium containing ammonium chloride. Yes, where the nitrogen, that is N15, is a heavy isotope of nitrogen. What is that? It is a heavy isotope of nitrogen. It is a heavy isotope of nitrogen. Since this nitrogen is a part of the DNA bases, every newly formed DNA molecule, it became heavy. And after several generations, what happened? All the bacterial DNA, it contained only heavy nitrogen. It contained only heavy nitrogen. And what is a heavy isotope? You may know about radioactive isotope, but what is a heavy isotope? See, N is here, they are mentioning N, right? This N is not radioactive. It is not radioactive. It is simply heavier than normal nitrogen. Normal nitrogen is N14. So heavy DNA can be separated from normal DNA using density gradient centrifugation. And then next step, the bacteria was transferred into a medium containing normal ammonium chloride. Normal ammonium chloride, in the sense the nitrogen, how much it will be? It is 14. Yes, and nitrogen, this normal is N14. And then samples now every new DNA strand would contain light nitrogen. Yes, now every DNA strand would contain light nitrogen, and the samples were collected after 20 minutes, 40 minutes, and later generations also. And this E. coli divides every 20 minutes, and each sample it represented one generation. And the last step is the centrifugation, and the DNA from each generation was extracted, and it was centrifuged using a cesium chloride, CsCl density gradient, right? And then let us see this diagrammatic part. So here you can able to see these are the centrifuge experimental result. So the centrifuge tube separates DNA according to density. So heavy DNA settles lower and the light DNA remains higher, and the hybrid DNA forms a middle band here in the middle. Okay, so this is because the heavier molecules move further during centrifugation. And then what happens in the generation? First generation is nothing but this is first generation 20 minutes. So what happens in first generation? Every DNA molecule contained one old heavy strand. It contained one old heavy strand and one new light strand. One old heavy strand and one new light strand. And all DNA showed intermediate, that is, it is like a hybrid density, and only one band appeared in the middle. You can able to see one band in the middle. And what happened after 40 minutes, that is the second generation. In the second generation, half of the DNA molecules became completely light. And here is you can able to see is here it is N14, so another N14. Yes, here is N15. Yes, you can able to see no N14, N15. So the remaining half was still contained one heavy strand. So therefore, two bands appeared. Two bands appeared. One is the hybrid DNA, which is having both 14 and 15, and also another one is a light DNA. So both were present in equal amounts. So if allowed to grow for 80 minutes, what will happen? See, this is a very interesting question. And since every generation doubles this light DNA, after 80 minutes, 4 generations will occur, and most DNA will become light. Approximately, we can say light DNA will be 87.5 percentage, and this hybrid DNA will be 12.5 percentage. And heavy DNA completely disappears. So what we can conclude from this experiment? What is the result of this experiment? See, the experiment clearly proved a DNA replication. DNA replication is semi-conservative. Say neither conservative nor dispersive models. It matched with the observed results. And one more experiment was performed. That is the Taylor's experiment. So it's not there. I'll explain you anyway. So a similar experiment was later performed by Taylor and his colleagues. So that too happened in the year 1958. So using a Visia faba, that is a broad bean. So instead of heavy nitrogen, they used radioactive thymidine in order to trace the newly synthesized DNA in chromosomes. So this results also confirm that chromosomal DNA replicates semi-conservatively. So next we are going to see the machinery and enzymes of replication. Replication cannot occur automatically. So many enzymes, they work together. So the most important enzyme is DNA dependent. DNA dependent DNA polymerase. And why it is called as a DNA dependent DNA polymerase? Because it uses DNA as the template. DNA as the template to produce another DNA strand, and that's why it is called as DNA dependent DNA polymerase. And what is the function of this one? Say its function is joining deoxyribonucleotides together. Its function is to join the deoxyribonucleotides together in order to synthesize a new DNA strand. And let's see the speed of this DNA polymerase. So in here they have given an E. coli. Yes, E. coli is Escherichia coli. So what is the genome size? Genome size in the base pass is 4.6 into 10 power 6 base pass. And what is the time taken? Time taken to replicate is 18 minutes. And also how many base pairs per second? 2000 base pairs per second. So this shows how incredibly efficient DNA polymerase is, right? And also what is the accuracy of this one? So the enzyme is not only fast. The enzyme is not only fast, It is extremely accurate. It is extremely accurate. Even a single mistake, it can produce mutation. So the DNA polymerase, it has very high fidelity and also energy source. So the replication, it requires enormous amount of energy. So where does this energy come from? It energy comes from deoxyribonucleoside triphosphates. Deoxyribo, here you can see deoxyribonucleoside triphosphate, that is DNTPs. So these molecules, they have two terminal high energy phosphate bonds similar to ATP. So this DNTPs, they perform two functions, one as the building block and another as the energy source. This dual role is a very important thing in NCERT syllabus. And why doesn't this DNA open completely? See, DNA molecules are extremely long, right? Yeah, they are very, very long. So separating the entire DNA at once, it would require enormous amount of energy. So only a small region opens at a time, and this small opening is called replication fork. So this small opening is called the replication fork. So next we are going to see the replication fork. So the replication fork is the Y-shaped region where DNA strands separate and new DNA is synthesized. And also it moves along the DNA as replication progresses. And why are two strands made differently? So DNA polymerase can synthesize DNA only in the 5' to 3' direction. So this creates a problem because the two template strands, yeah, it runs in opposite direction. They are anti-parallel. Yes, see the strands, yeah, they are anti-parallel. And also continuous synthesis, you can able to see where in the leading strand. So one template strand, yes, one template. So this is one template strand. It has a 3', yeah, to 5' direction. So DNA polymerase can move smoothly along this template strand, producing the new strand continuously. And this strand is called leading strand. And the next one is the discontinuous synthesis. Here you can able to see the discontinuous synthesis. So the other template strand, it runs from 5' to 3'. And since this DNA polymerase still works from only 5' to 3', it cannot copy this strand continuously. So instead, it synthesizes short DNA pieces. So these are the short DNA pieces. They are called as Okazaki fragments. These are called as the Okazaki fragments. So these fragments together, they form the lagging strand. Then what is this DNA ligase? What is the function of this DNA ligase? See, after all the fragments are synthesized, another enzyme, that is the DNA ligase, it joins the Okazaki fragments into one continuous DNA strand. So it joins the Okazaki fragments into one continuous DNA strand. So without DNA ligase, the lagging strand would remain broken into pieces. Let's see this diagram. You can able to see a Y-shaped replication fork, right? It shows the two template strands. You can able to see on one strand, that is on the one side, the leading strand is synthesized continuously toward the replication fork. On the other side, the lagging strand is synthesized away from the fork in short Okazaki fragments. And these fragments, they are joined by DNA ligase and produces a continuous strand. And let's see the origin of replication. See, replication does not begin randomly. So there is a specific DNA sequence where this replication starts, right? So the site we called as the origin of replication, ORI. Okay, in E. coli, this replication, it begins at a single origin. Where it begins at the single origin. And this concept is also important in recombinant DNA technology because vectors, it should contain an origin of replication in order to allow inserted DNA to replicate. And let's see this replication in eukaryotes. So in eukaryotic cell, DNA replication occurs during the S phase. So S phase is nothing but the synthesis phase of the cell cycle. So here the replication and cell division must be tightly coordinated. So if DNA replicates, but the cell fails to divide also, the chromosome number doubles, it leading to polyploidy, which is a chromosomal anomaly. So we have come to the conclusion part. So let's have a quick revision. Let's see here, semi-conservative. So each daughter cell, so it is having one old strand as well as one new strand. And Watson and Crick, so base pairing suggests a copying mechanism. And Meselson and Stahl, so they proved the semi-conservative replication, yes, using these heavy isotopes and also template strand. So one old strand that guides the new strand synthesis, and also the DNA polymerase. It is a main enzyme. It polymerases 5' to 3' ends, and DNTPs, it acts as a substrate and energy for polymerization. Replication 4 is a small opening where the helix is copied, and continuous or discontinuous, it leading made whole and also lagging in fragments. DNA ligase joins the discontinuous fragments, and origin of replication, so it's a definite start state only and also supplied by the vectors. So we have concluded that parts, children. So watch the full video. Don't skip the video. Watch it fully. We'll see you in the next video. Thank you.